Good afternoon, everyone. Hope you're having a great Tuesday. We are going to jump right back in where we were yesterday. If you remember yesterday, what we were talking about was proteins, and we were talking about, well, what we're going to be continuing on is the structure of proteins. Yesterday, what we did was we went over the primary and secondary structure, which the primary structure is stabilized by covalent bonds, a peptide bond being a specific example of a covalent bond, whereas your secondary structure is stabilized by backbone hydrogen bonding. Now, we touched on a little bit yesterday the tertiary structure, but today we're going to get a bit more into that. And we're going to couple that with a couple of examples. Um, and hopefully by the end of today, we will be into a portion of our discussion of myoglobin and hemoglobin where we're talking about protein function. So, with that all ready to go, I'm going to expand my PowerPoint. There we go. That's full screen. Now I'm going to share my screen coming right up here. Okay. So, whenever we talk about protein structure, there's a couple of different models that are kind of the conventions to use. Um, when it comes to protein structure, it's kind of simplified, I guess you could say, to talk about muscle proteins. Now, muscle proteins, think about protein structure as kind of a big umbrella. Underneath that umbrella is globular proteins and filamentous or fibrous proteins. The filamentous or fibrous proteins are commonly known as your structural proteins. Now, one thing that's important to remember is that despite those being your structural proteins, it is an area of interest for people to know about the structure of other proteins. And that goes for all proteins for that matter. So, your muscle proteins being your filamentous or fibrous proteins, there's a couple of properties and traits about them that is pretty well recognized as being universal. One thing is that they are very strong. Another thing is that they have very repetitive sequences. And these repetitive sequences, whenever I say that, I'm going back to the primary structure, which is stabilized by covalent bonds. I mean, there's a repetitive pattern, and that pattern might be, three amino acids, three unique amino acids like lysine, arginine, alanine, repeated over and over again, or that pattern might be a little bit longer and it might be something like lysine, arginine, then a break of 10 amino acids, then that same lysine, arginine sort of thing. So there's some repetition and there's patterns and there's things that people recognize about them. Now, the key is for the significance of that highly repetitive sequences is it makes them kind of boring. Um, but that's kind of like saying that roads and bridges are boring. They're essential. I mean, to me, that's not very interesting, but I do want to be able to take for granted that I can drive on a bridge that will not collapse whenever I drive over it. Okay, so your fibrous proteins, that's what they are. Globular proteins are a little bit more, I would, I feel comfortable saying they're more interesting because of the fact that they're so diverse. There's so many varieties of globular proteins. Now, we'll talk about muscle fibers because they give us a good model for understanding structural proteins. So, muscles are composed of bundles of single large cells. Now, your myofibrils are what are the muscle fiber components. So if we look, it's important to look at the individual units. And I'm going to go through all this, but then I want to go over to this model on the right-hand side. They're composed primarily of proteins, actin and myosin. Those are not the only proteins, but those are the most common proteins and the most relevant proteins. Now, with that said, this right here is a microscope image. And what that's showing is, you can see, you can pick out a couple of structures. Now, these structures right here have unique names. For instance, this barrier over here is known as the Z disk. This barrier right here is known as the M disk, And then over here, we've got another Z disc. Now, what these are is looking at individual, I'm going to jump back here, the individual myofibrils or the individual sarcomeres. So here is a sarcomere, and think about that as like the base unit that is kind of merged together to make a muscle fiber. Okay. So that sarcomere has boundaries known as Z discs. In the middle, there is an M disc. Now, the significance of this is basically in order to do this, in order for me to move my fingers, in order for me to flex and everything like that, it's all these sarcomeres that expand and contract, expand and contract. Now, how is that taking place? Well, that action is taking place with the help of the way that actin and myosin interact with one another. Okay, so understanding that this sarcomere, what we're looking at is the individual thing that helps me flex my muscles. Okay, now to look at kind of the regions of this, there are a couple of different bands. Those bands are known as the I band. So this I band right here is nothing but actin filaments, thin actin filaments. So this purple or kind of magenta colored space right there. Oh, I'm sorry, I'm seeing chat. I'm sorry, but where are these slides? They so these slides they are from Monday. Oh, second. Okay. Thank you very much for taking care of your classmate, everyone. I apologize for interrupting myself then. It's okay, Joel. Don't worry about it. So the I band, that is just actin filaments. And if you look at that, the way that I always kind of think about it is the I band is anchored at the Z disc. And this is a little bit alphabet soup, but bear with me. Now, the other major component is the A bands. Okay, so the A bands are kind of opposite, but they work together with the I bands. Now, the difference is that the A bands, oh, not that. Um, the A bands are what are anchored on the M disc. So I comes from Z and M, or sorry, A comes from the M disc. Our two discs are where proteins kind of go outward from it. Now, with that, there are kind of regions within these sarcomeres. Now, the trickiest part about this is to know which regions kind of shrink and which regions grow. So, if you look at the Z discs, when a um, when a muscle cell contracts, the Z discs get closer together. When a muscle cell expands, the Z discs grow farther apart. Okay. Now, what are the implications of that? Well, the implications are the I band and the A band will change their size, as will the H zone. Now, the H zone is nothing but myosin. Okay, so thinking about, let's just look at the H zone, that middle region that is nothing but myosin. When the Z discs get closer together, what do you think the H zone is going to do? And your options are it can either grow or it can shrink. When these Z discs get closer together, what does the H zone do? It's going to shrink. You're absolutely right. It's going to shrink because basically this right here, that protein right there, which is none other than actin, actin is going to move closer to my M disc. It's not going to elongate, but instead it's going to get closer because of the fact that the Z discs are contracting and they are getting closer together. Now, what that also means, since our H zone is getting smaller, that means that our I band, notice the I band is nothing but actin filaments coming from the Z disc. What do you think is going to happen to the I band? Is that going to grow or is that going to shrink whenever the Z disks get closer to the M disk? That too is going to shrink. You're absolutely right. Now, one region that will grow in all of this is the A bands. Because if you look at the A band region, the A band comprises the H zone, which is just myosin, but it also contains this overlapping region right here. So that overlapping region is where we have both actin and myosin interacting with one another. Now, what I want you to think about as, okay, what are my takeaways other than this is a bunch of letters? Your takeaways are, or the important takeaways that I want you to have is that your boundaries are the Z disks. Actin and myosin will interact with one another, and that is how muscles expand and muscles contract. Now, in terms of the bands and the zones and everything like that, I don't want you to get lost in that. Instead, what I want you to think about is actin and myosin, their interaction with one another, and the Z disks and M disks. So I want you to say, okay, from my Z disks, that's where my actin filaments go out. From my M disk, my middle disk, that is where my myosin filaments go out. If you can do that, that's going to be kind of that would hit the high points of the questions that I'm going to ask you about the structure of an individual sarcomere. Now, there are other proteins that are involved here. The other proteins that are involved, and actually before we get to those other proteins, what I'd want you to do is looking at this structure right here, or looking at this model. I'm going to scribble out this one so you don't get lost on that. What we have is a Z disc. And here is a thin actin filament projecting outward toward that M disc or that M line. And we've got two of them. We've got two of them from each Z disc. So within that overlapping region, which we know as the part of the A band, what we have is myosin and actin proteins interacting with one another. Now, a fair question is, well, what keeps those things intact? And we're going to get into the movement here in just a little bit, but what keeps these things intact? Well, there's a couple of other proteins. We've got two other major proteins. One is titin. Every time I see that, I always think of the old French comic book Tintin, which I remember my aunt and uncle got me for Christmas a long time ago. I always see titin. Titin basically connects the M disk or the M disks to the Z disks. So molecules of titin extend from the Z disk to the M line and act as springs of sorts, which is why we've got this kind of spiral structure there to keep myosin filaments centered within the sarcomere. So they connect the Z and the M disks to one another, and they keep myosin basically on the tracks. Now, another protein known as nebulin extends from the Z disks and are thought to determine the length, or the appropriate length of the actin filaments. So essentially, titin and nebulin are supporters of myosin and actin. Titin facilitates or helps out myosin, whereas nebulin helps out actin. That's effectively what we've got going on here. Now, the important part about all of this is if you look, I want you to recognize the differences between this structure on top and this structure on bottom. The structure on top, we've got all of those myosin head groups. And what you notice about those head groups is they are interacting with the actin filaments. Now, these two models are not necessarily shown to be like reductive or to show you like, hey, well, we're adding pieces in, but instead to show you, well, the interaction between myosin head groups and actin is going to be something that can take place and can release. So it's like they can anchor, but then they can release. Whenever that happens, well, then that enables the muscles to either contract or relax, or contract or expand. Nope. Yeah, contract or expand. Now, what myosin looks like is this is basically a myofibril or a myosin um, head group interacting with an actin filament. The myosin head group is going to grab on to the actin filament and release. Grab on, release, grab on, release. Now, what that's going to look like is what we're going to see on the next slide. This is a sarcomere. This is another sarcomere. This is in its most expanded or most relaxed state. And then down here at the bottom, we've got our most relaxed state. And what we're seeing is these actin filaments. So let's track this going from 1 to 4. This is an actin filament that I'm putting a circle around. And then we've got that contraction taking place. At some point, our two actin filaments anchored at opposite Z disks overlap one another. So we've got this region right here where two actin filaments overlap with our myosin filaments. So that's where it's very tightly contracted. Now, how this ultimately relates to kind of physiology and what are the steps taking place here? The steps that I want you to know about is that myosin head groups grab onto actin filaments. And during muscle contraction, each of those sarcomeres is going to get shorter, where the Z discs are going to get closer to one another, but they're also going to get closer to the M disc. Visually, there's no change in the width of the A band, but both the I bands and the H zone completely disappear whenever it's completely contracted. This is caused by actin and myosin, and thus the actin filaments move into the A band and H zone, and that's how they basically disappear. Now, this is great from like a 10,000 foot view, but what about a kind of mechanistic, or what about a mechanism that's taking place there? Well, here's the steps that are taking place. So, starting up in the left-hand corner, we've got part of a myosin molecule, a myosin head group here, is bound to an actin filament. And what's going to happen to this myosin head group is it's going to bind the molecule ATP. Now, I've mentioned ATP previously. We mentioned it in the context of the mitochondria. ATP is your energy currency. Now, what's going to happen here is your myosin head group, which is bound to an actin filament, is going to, just one second, let me jump back. Um, your myosin head group, which is bound to the actin filament, is going to bind ATP. Whenever the myosin head group binds ATP, then it's going to release from the actin filament. Now, I think everyone in this class can agree that that release is, well, there's a change. That interaction is no longer taking place. A fair question is why? I mean, you've got the surface explanation of, oh, well, it bound to this new molecule. But when something binds to another molecule, I always kind of think about it like my hand. Here's the shape of my hand. When my hand binds to a pen, the shape of my hand has changed. The shape of that myosin head group has changed. In which case, let's say that I'm holding onto this thing, and then whenever I bind onto my pencil, I have to release that other thing. So the other thing that I was alluding to was actin. Whenever I bound to this new thing, being my ATP or adenosine triphosphate, well, I can no longer hold on to the actin filament. Okay, now what's going to happen here is that when the actin filament is released, well, my hand or the myosin head group is going to undergo another change. Not only has it lost its affinity for that that actin filament, but it basically goes like this. And whenever it does this motion, now all of a sudden it can pick up, and let's see, I don't have a good figure here, but it can bind to another part of actin. So it binds ATP, releases actin, and now it kind of lurches forward. And whenever it does that lurch forward movement or motion, movement, whatever, it's going to take ATP, our high energy compound, and break it into ADP and inorganic phosphate. Now, we haven't talked about nucleotides, but what I want you to know, and the need that I have for you to know about this right here is ATP is basically the high energy compound. ADP plus PI, this reaction right here is showing that basically we've cashed in that high energy compound. We've broken off. It's almost like we bit into the piece of candy of sorts. Because this reaction, I'm going to rate RXN, releases a huge amount of energy. Now, in addition to releasing that energy, now all of a sudden, our myosin head group, well, it's no longer binding ATP, but instead it's binding ADP and PI. So it's almost like it's beginning to release that. At that point, it can then bind onto a new part of actin. Or, yeah, of actin. And whenever it does that, well, it's going to release the remaining ADP. And where are we again? We're back where we started. So basically, the way that I always kind of think about it, and bear with me with this example, um, imagine that the myosin head group is basically what it's doing is it's kind of like as you watch my face in this the cell, it's like it's doing this. Hump, 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 hump. It's Homer Simpson eating like a 10-foot long Subway sandwich. And just so you guys know, I get $5 every time I mention Subway. So that's 10 bones right there. Not really. I haven't been to a Subway in 20 years. Um, but effectively, think about myosin as being something that it binds, it's bound to actin, releases, so it can reconnect, release to reconnect, release to reconnect. And the way that it does that, the way that that motion is taking place is with the catalyzing this reaction of ADP to ATP, or sorry, ATP to ADP and PI. A more formal representation of that is shown right here. This has the details of what's taking place, where we have this lurching, this movement forward. And what that's doing is that is pulling the actin filament closer, in which case that is the contraction, getting those Z disks closer to one another. Okay, so that's essentially what's happening in the case of a movement of a muscle protein or a muscle cell or a muscle filament. We have these proteins that are very structured, they're very linear, they don't have a lot of elaborate shape to them or anything like that. Interaction, bind, interaction, bind. Okay, now um, calcium is going to have an influence on those because calcium can bind to them, as can other proteins. So, you know what? We're going to move past that. Okay. So I want you to know about the sequence of events that's taking place for structural proteins or for your filamentous proteins. I want you to know about I want you to know about myosin and actin, how those filaments interact. Knowing this sequence of events is something that I do want you to know about. Or sorry, this sequence of events is something that I want you to know about. Now, when it comes to protein structure and talking about globular proteins, arguably it's a little bit more interesting. Globular proteins don't have an expected pattern necessarily. Um, And what I mean by that is they've got a lot of diversity of amino acids. The primary structure of a globular protein is going to be considerably different than the primary structure of a filamentous or fibrous protein. A filamentous or fibrous protein might be like four amino acids on repeat, whereas a globular protein, if it's 400 amino acids, it's going to use all 20 proteinogenic amino acids. And it's going to have a region that's highly polar, a bunch of regions that are highly nonpolar. Now, due to this kind of diversity and due to the range of different amino acids, that makes it difficult to predict the structure of a protein, of a globular protein. And I kind of alluded to the importance of being able to do that because if you understand or can predict the structure of a protein, then one thing that can be done is a drug could be designed for it if there's that concern. Now, with that said, whenever we talk about globular proteins, there's basically two states, a folded and an unfolded state. Now, to give you a little bit of a reminder, think about this is one polypeptide, and we've got an N termini and a C termini. Now, I've taught this class a number of times, and I think that one thing that I've noticed over the years is sometimes people have this like disconnect. This molecule right here that or actually the molecule that's shown here in the unfolded state and folded state, it's got an N termini and it's got a C termini. And let's say that this is 600 amino acids. Amino acid number one might be aspartic acid. Amino acid number two might be a glycine. Amino acid number three might be a glycine. All of those amino acid structures, all those proteinogenic amino acids, well, they make up this molecule right here that I'm showing in the unfolded state. Then what happens is in an aqueous environment, that molecule made up of all of those amino acids will take on this kind of crazy wonky shape right here. Now, what you see right here, and kind of a comparison of these two, is on the left-hand side in its unfolded state, you basically, all that you've got is the primary structure. On the right, in its folded state, well, this protein does have a primary structure. Absolutely, no doubt about it. Does anyone see any secondary structural elements in the folded state? And if so, add to the chat what you see. An alpha helix. Absolutely. It's a little bit, it's always, the helices, I think, are always like, oh, yeah, I see that. The beta sheets are a little bit trickier, but here is one, or here is a beta strand because we've got that kind of ribbon arrow shape to it. And that's pretty common for what you'd see. Now, what that means is that this protein in its folded state has a couple of things. It's got an alpha helix, it's got a beta strand, and then this right here, I always feel like this is a little bit intuitive. The flat ribbon, yes, the flat ribbon is a beta strand. It's kind of tough to see, but you basically got two of them. This one and then this one right here, kind of shielded by that alpha helix. So that's two beta strands. Um, where I've drawn my arrow, I've asked this question in the past, um, and the person who has the answer is typically very reluctant to say it because they're like, certainly that can't be it. But I like to ask, what would you call this part? See this green line. Um, I certainly wouldn't call it an alpha helix. I certainly wouldn't call it a beta strand because I mean, heck, it doesn't look like either of those. I mean, I think that I would call it a random coil. And I hate to disappoint you guys, but yeah, that's what it's called. It's just considered a random coil because there's no organization or order to that structure. So essentially, now with that said, is a random coil a secondary structure? Not really, because a random coil is just that. It's totally random. It's all over the place. There's no organization to it compared to an alpha helix. An alpha helix has a pattern. A beta strand has a pattern. They're both stabilized by backbone hydrogen bonding, whereas a random coil isn't going to be stabilized by backbone hydrogen bonding. So this folded structure right here, well, this is the tertiary structure of this protein. We've got secondary structural elements, the alpha helices and the beta sheets that basic or the beta strands that kind of, well, they position themselves completely to give us that folded structure. So when you look at the unfolded structure of a protein, in that case, it's just a bunch of random coils. But that's basically only your primary structure. The folded structure of a protein includes your primary as well as secondary structures. But that tertiary structure, that whole folded shape is not just dictated by um or sorry, that folded shape is going to be dictated by R group interactions, hydrogen bonding between an aspartic acid or an arginine and an alanine, or not an arginine and alanine, but R group interactions, how those different amino acid R groups interact with one another. Now, a fair question is, well, how do we go from that folded state to the unfolded state? And the answer to that is with a process known as denaturation. And denaturation is a process by which you basically break all the interactions that take place between R groups that exist in that folded 3D shape. Now, to put that into perspective, this is a single polypeptide right here. That single polypeptide is going to be stabilized by hydrophobic interactions, hydrogen bonds, ionic interactions, dipole-dipole. So there's any number of interactions that are going to take place there. Now, with that in mind, and this is just kind of a primer, um, hydrophobic interactions. Let's think about the class of amino acids that are going to be interacting with one another in a hydrophobic interaction. You're going to have your non-polar amino acids interacting with one another in a hydrophobic interaction because they hate water. And in those hydrophobic interactions, they can be happy as a clam, hating water. Now, with that in mind, I think that I've kind of casually referenced this sort of question before. When you have a hydrophobic interaction, or when you have hydrophobic interactions, um, are they going to be influenced by changes in pH? Hydrophobic interactions, they detest water, so they'll force water out of there. If you add more protons to the situation, are these non-polar groups going to be influenced by that? And whenever I say non-polar groups, think about something like this. If we have an interaction between CH3 and CH3, is water going to have anything to do with that? No, because they're both like, nope, we don't like water. Stay away. Now, what would be kind of the carrier of a proton? These guys are going to have the same attitude of, well, we didn't like your brother. We don't like you either. Sorry, H3O or hydronium, you're basically the same thing. So, what I'm getting at there is a hydrophobic interaction is not going to be influenced by a change in pH. And my point there is, how do we go from this state right here to this, where we disrupt all of these different types of interactions? Well, one way that can be done is by heat. This is commonly known as heat denaturation. Another way is going to be chemical denaturation, and we're going to talk about some of the denaturants of that and that you use for that in just a little bit. So, when it comes to, well, which of these structures kind of matters? Your fully folded and or your fully folded form of a protein is the functional part of a protein or a functional form of a protein. This one on the left, this is the one that's ready to carry out a catalytic reaction. This is the form of hemoglobin that can bind oxygen. This one on the right-hand side, after it's been heated up, can't do anything. Which you have some examples of heat denaturation that are a little bit accessible where you see a change. One example of heat denaturation that you're probably familiar with is when you make fried eggs. You're denaturing all of the proteins that constitute the egg white, and then you're denaturing proteins in the yolk as well with a bunch of cholesterol in there. So you're denaturing proteins, and what happens to that egg, or the white of an egg? Well, it gets less liquidy. It becomes a solid. It becomes a solid network that, well, it takes more force to pull it apart. Okay, now what are the stabilizing forces? And I kind of alluded to this a little bit ago, but what are the stabilizing forces in the tertiary structure of a protein? R group interactions. Well, tertiary structures, right here. Hydrophobic bonds, hydrophobic interactions, where we've got amino acids like phenylalanine, isoleucine, and valine, all your non-polar residues. We've got ionic interactions between amino acids like lysine and aspartic acid. So K and D. Now, this one's a little bit trickier, but metallic bonds. In order for a metallic bond to take place, well, you might have to have something that directly binds to that metal ion, in which case you'd probably have to have something like an aspartic acid or a glutamic acid, because those ions are going to kind of coordinate that metal. Then last but not least, this guy right here. Could I get somebody in the chat to tell me which amino acid do they think is responsible for forming a disulfide bond? And I'll give you a little bit of a clue. Cysteine and methionine are the only two amino acids with sulfur in them. One of them is nonpolar and one of them is polar. Cysteine. Wonderful. Great job. Cysteine is the amino acid that forms a disulfide bond. Now, with that said, just as like a kind of trigger of sorts for me, I always just write CYS, CYS. In addition to that, one of the reasons that I remember it's cysteine that does this is because if I think about methionine, well, methionine looks a little bit like this. This is the R group. It's got that methyl group there. If I removed that methyl group, well then, yeah, I could. But that methyl group, it's not going anywhere. So whenever you're asked about disulfide bonds, if you're tempted to go, oh, it's either cysteine or methionine, just think about the names and say oh yeah, yeah, that methyl group there, that's going to block me. Now, over here on the right-hand side, this is just another illustration that we all know secondary structures are stabilized by backbone hydrogen bonds. There's the alpha helix, there's the beta sheet. Okay, so R group interactions are the key determinant of tertiary structure. They're also influential in quaternary structure, but when we talk about tertiary structure, we're talking about a single polypeptide. Just a second. Okay. R group interactions. Hydrophobic interactions, these up here in the middle, your nonpolar residues, despite them being kind of weak interactions, they are so angrily against water that they pack super well together. Typically, hydrophobic interactions are going to stabilize the core of a protein. So, kind of in the middle of a protein, which, if I jump back, in the middle of a tertiary structure of a protein, like let's imagine that this molecule right here, we could rotate it and look at like the center of that protein. It would be a bunch of non-polar residues that cluster together to basically keep everything else out. So your non-polar or your hydrophobic residues are going to pack into the core. Hydrogen bonding, well, it's going to stabilize polar regions of a protein. Now, I like to bring up these two first because I always like to think back to or I like to think about these two types of interactions as being relatively weak. If you compare hydrogen hydrophobic interactions to ionic bonds, well, ionic bonds are super strong. Disulfide bonds, when you look at those, a disulfide bond, this is the one covalent bond that we talk about with respect to the one covalent bond that we talk about with respect to stabilizing tertiary and quaternary structure. So ionic bonds and disulfide bonds are considerably stronger than hydrophobic and hydrogen interactions. However, when we talk about protein structure, it's very important that you think about hydrophobic and hydrophobic bonds and hydrophobic interactions and hydrogen bonds. The reason is, to me, it's a good way to think back to the Disney Pixar movie A Bug's Life. If you've ever seen A Bug's Life, there's a scene where Hopper, who is the leader of the grasshoppers, is with his brother and they are at a grasshopper bar of sorts. And his brother says something like, we don't I don't think we need to go back to the ant colony. I think we're good. Let's not. And Hopper says, "No, we need to." And a I don't know if it's a complex movie. Oh, yeah, it's a complex movie. But he basically says, no, we need to go back there because we need to assert our dominance on the ants. We need to show them that they work for us. And he uses this, he throws a piece of food at his brother and he says, does that hurt? And he's like, no, it doesn't. And then he releases thousands of pieces of food at his brother. And he's like, well, does that hurt? Actually, it wasn't his brother, but he releases all that and he's like, does that hurt? And you can't see the guy. My point is, the individual hydrogen bond is not particularly strong. But when you have thousands upon millions of hydrogen bonds, that's going to stabilize a structure. Same thing with hydrophobic interactions. The individuals are weak, but in mass, they're tremendously strong and they're a driving force of folding the protein. So ionic bonds, disulfide bonds, hydrophobic and hydrogen bonds are all going to stabilize, but to varying strengths. And they all need to be accounted for and thought about, like what types of or which amino acids are going to interact in these. Now, to fill in this, to kind of treat this like a table, I think that it's worthwhile to kind of have some amino acids that you can pull from and say, okay, well, these two residues will interact and give me this type of interaction. Hydrophobic interaction. Well, valine, which residues will do this? Valine and isoleucine. If I have those two interact with one another, hydrophobic interaction. Then hydrogen bond, serine, and we'll make it easy on ourselves and we'll say threonine. Those two can interact with one another. Serine and threonine, they both have a hydroxyl group on the R group. Those two will interact. Ionic bonds. Well, here we want to pick one amino acid that can be a cation and one that can be an anion. Well, our polar positive residues like histidine and a polar negative residue like glutamic acid. Disulfide bonds, well, that is just cysteine. Or, well, just cysteine and cysteine. I think that it's good to have this sort of combination to pull from or to change it. And I'm going to erase all of these. So if you want to take a snapshot of this, go ahead and use Snagit to capture it. Um, or it's here, it's on the video. Um, I think that it's also worthwhile to think about it in terms of groups. We need a nonpolar plus a nonpolar to interact for hydrophobic. Hydrogen bonds. We need a polar uncharged is our best option. Polar uncharged. Two polar uncharged residues to interact. Ionic, well, we need a polar positive, PP, and a polar negative, PN. Those are going to be the easiest go-tos. Now, is this an absolutely comprehensive list? No, but I would say that this gets you about 90% of the interactions. Does everyone understand my coding? Give me a number if you understand my coding, and then a letter if I'm confused. Okay, great. Happy to hear that or read that. Okay, so all of this is leading to, and what we're talking about is R group interactions. This is one of the reasons that I wanted to introduce amino acids at the end of last week, because I wanted you guys to be looking at those structures and spend more time with them. Now, these are determinants of protein structure, tertiary structure. I got a message of, could you say what the letter stands for? Okay, sure. NP is nonpolar, and then PU is polar uncharged. And then PP is, I'll just say polar positive. And then PN is polar negative. Okay. Now, disrupting these interactions. That's what we have to do in order to denature a protein. So we use different denaturants. These are chemicals or treatments, you could say, that interrupt those interactions. So there are two common reactants that are used for breaking disulfide bonds. They are known as mercaptoethanol and dithiothriotol. This mercaptoethanol is going to, excuse me, also be beta mercaptoethanol, also known as BME, beta mercaptoethanol. Dithiothriotol, also known as DTT, what these two reactants do is they disrupt disulfide bonds. So they break a covalent bond. So they break disulfide bonds. What that means is that if you had a protein and you heated it up and you did a bunch of other stuff to it, if you didn't add dithiothriotol or beta mercaptoethanol, you're not going to break that disulfide bond. Because think about it like this: if you can break a covalent bond with heat, what's that going to do to the primary structure of your protein? It's going to break every one of those bonds. So if you heat, basically, if you heat the protein, or if you were to heat a protein to a high enough temperature to break a disulfide bond, you would have heated it up enough to break a carbon-carbon bond or a carbon-oxygen bond. So, if you were asked the question, can heat break a covalent bond? Yeah, it can, but it's not going to be super selective. Whereas beta mercaptoethanol or DTT both specifically break disulfide bonds. Now, next up, detergents. Detergents are chemical compounds that disrupt hydrophobic interactions. So breaking up those nonpolar interactions. So if we jump back, if you used a detergent, you're going to break nonpolar interactions. Great. Heat. Well, heat is going to be effective for breaking hydrogen bonding. So if you used heat and a detergent, you're going to disrupt the core of the protein. You're also going to disrupt some R group or some R groups within the polar region of your protein, or any polar region within your protein. If you were to adjust the pH, you can alter hydrogen bonds as well as ionic interactions. Think about it like this. If you've got, if we jump back here to this sort of ionic bond, if you've got an arginine and let's say a aspartic acid, if they are interacting with one another and you make the environment extremely acidic, and this COO minus, I see in just a second, you get this COOH. Does COOH have a positive charge? Nope. So it's not going to participate in ionic interaction. I've got a question here. Go ahead.
Hi, sorry, I didn't want to interrupt, but for me, for like heat, like we were just talking about, I guess my question is it's not selective, right? Like it depends on the amount, but it can break all the bonds. So am I thinking about it too generally?
So, um, no, you are not. You're fine in the way that you're thinking about it. The heat is going to be non-selective, but the magnitude of it. Think about it like this. If we heat it, typically whenever we're aiming to break hydrogen bonds, we're basically going to heat up to say, 95, 99 degrees centigrade or Celsius. So that's not such an extreme heat and duration that we break all the other bonds or break peptide bonds, for instance.
So maybe the way to think about it is like heat is selective at like a lower grade.
Perfect. Perfect.
Okay.
Yeah, great. I like that. Thank you very much. That's fantastic.
Okay, thank you. I just want to make sure I got it right or was understanding it correctly.
Sure. So I'll add to this within reason. Within reason. Okay. So pH can adjust that. Then we've got chelators, which chelators are molecules that bind metal ions. Now, I remember hearing about this a number of years ago. There's a treatment, like if anyone were to, I don't know if it works for lead necessarily, but for heavy metal poisoning, um, EDTA is a compound that basically what it does is, and under a doctor's supervision, chelation therapy is a way that EDTA and other compounds will basically be injected into your body. And essentially what they'll do is they'll kind of like encapsulate a metal ion. And so that's a way in which it's used in like a clinical setting. But in terms of protein folding, if we've got a metal ion that is interacting between a pair of amino acids and then something comes and just scoops it out, well, then that metal ion isn't going to be interacting between those two amino acids. So it's going to disrupt that interaction. Then, last but not least, urea and guanidinium hydrochloride or guanidinium chloride. These are compounds that are known as hygroscopic reagents. These are chemicals that disrupt hydrogen bonds. These are also known as chaotropes or chaotropic agents. And this is a substance, or these are substances that basically they effectively do the same thing as the heat would do. They're going to disrupt hydrogen bonds, in which case, we go back, they would address these polar uncharged reaction interactions. They're going to disrupt those, the threonine interacting with the serine, and so on. So all of these denaturants do different things. I think that it's helpful to think about, well, in application, what is the aim to do? If there's one thing that I would say, make sure you know this above all else, this one right here. Beta mercaptoethanol and thiothiotol. Make sure that you know those because those are kind of unique for a specific unique amino acid interaction. The rest of them, I think that you can kind of use logic to approach. Changing pH, is that going to impact two nonpolar amino acids from interacting? No, not really, because I know nonpolar interactions are hydrophobic. Now, Where and why is all of this important? This is important because, again, we want to know how a protein folds. We want to know how we can take the primary structure of a protein and get this thing on the right. How do we go from this thing on the left to the thing on the right? And sure, within a cell, it kind of organically or naturally happens, but how can we predict that this protein on the left is going to look like that thing on the right? Well, we talked yesterday about how Chow and Fastman did secondary structural analysis. And they said, here's how we can predict if an alpha helix or a beta sheet's going to form. This fellow on the right-hand side, his name is Christian Anfinsen. I'll write his last name, Anfinsen. What he did was he studied a protein known as RNase. This is a pretty well-known protein or class of proteins. And what it does is it takes RNA and chews it up. It chews up an RNA molecule into individual nucleotides. Now, if you've ever heard of RNase, you've possibly heard that there's different RNases. And what I mean by that is there's something known as RNase A, and there's RNase H. They all have slight variations on what they kind of chew up, but in a general sense, ribonuclease is going to take an RNA molecule and chew it up into individual pieces. So this is a protein. Oh, sorry, I heard the front door open and my kids start yelling. So that means they're home. So I had to fortify myself and close the door because otherwise it's going to be loud. Um, so ribonuclease is this enzyme that takes RNA and chews it up. It's a pretty well characterized protein. Even back into the 1950s, people were familiar with and studying it. And Christian Anfinsen was a structural biologist or structural biochemist. So he was interested in it basically like, what does this thing look like? Because the more I know about what this thing looks like, the better I understand it and the better, maybe we can manipulate it. So he was studying it kind of as a model of what does a protein look like in 3 dimensions. And what he did was he took two solutions, 8 molar urea. Urea is one of our chaotropic agents. It's going to break up hydrogen bonds. It's going to break up almost everything else along the way. Then he also used beta mercaptoethanol. And that beta mercaptoethanol was to break disulfide bonds. What this shows on the left-hand side is the kind of semi-folded structure of ribonuclease. This, 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 and this are all disulfide bonds. So what that means is looking at this, we can see that at position number 40, there's a cysteine. At position 95, it's also a cysteine. A disulfide bond is formed between those two amino acids. So he did have to use beta mercaptoethanol to fully denature this protein. And he did just that. He was able to add this 8 molar urea and the beta mercaptoethanol and get the unfolded protein. Then what he did, you look at my arrows here. It's important. It's not just a matter of, oh, well, I'm going to draw the arrow this way and I might as well draw it that way. What Christian Anfinsen's work continued to do was, all right, well, let's see if we can dilute out that urea and dilute out that beta mercaptoethanol and see what happens. And lo and behold, when he diluted out those two substances, ribonuclease went back to its native folded structure. So essentially what that means is you can denature and renature a protein. The significance of that is everything, I feel like this is kind of a metaphor for life, everything that was inside this protein, the primary structure of this protein, was enough for it to get to this native folded state. What that means is there's no special sauce, there's no magic that's sprinkled onto a protein as it goes from its denatured state to its fully native active state or its folded state. And that was a pretty big deal because essentially what that says is, well, if you know the primary structure of a protein, it's going to get to the native state. And essentially what that means is that, and all of this is done in a test tube or in test tubes, probably not just one, but the removal of those denaturants, the urea and the beta-mercaptoethanol, was enough to get ribonuclease to be in its folded state. That was, I would say, maybe not the first, but that was um, an early cue that we as a, group of scientists could predict the folded shape of a protein from the primary structure. So connecting the primary structure to the tertiary structure. Now, with that said, I want to kind of highlight a couple of terms. Native, I've used that term, and that is the common term used to describe the properly folded structure of a protein. Now, in addition to that, denatured that's your unfolded form of your protein. So that's when you've taken a protein and basically made all of it a random coil. There's basically no R group interactions. This is the form of the protein that's not going to do anything. Instead, it's just like, okay, cool, it's a linear polypeptide. Now, with that in mind, this is where we're going to begin talking about functions of proteins. And it's important to know what a protein domain is. So, protein domains are kind of regions of a protein. These are independently folding regions of a protein that often have specific functions. They do something in particular. Characteristics of them is that they can evolve, function, and exist independently. They're typically 25 to 500 amino acids and are found in multiple proteins across different organisms. Now, domains can be a couple of different things. They can be catalytic, they can be binding, or they can be structural. For a catalytic and binding domain, I want you to think about this as function. They have a particular function. For example, is anyone familiar with glycolysis? Glycolysis is a process where we take basically glucose and we make ATP. We make a bunch of it. And that's entirely too simple of an explanation. But along the way, there are a bunch of enzymes or proteins involved in this process. One of the things that they do is they bind ATP. The first enzyme in glycolysis is an enzyme known as hexokinase. Hexokinase is the enzyme that one of the things that it does is it binds glucose and it binds ATP. Okay? I want you to remember that. And I want you to now look at this right here. This myosin head group. The myosin head group is a bunch of amino acids. Whole bunch of them. And this particular structural region of the myosin head group binds ATP. So functionally, part of the myosin head group and part of hexokinase do the exact same thing. Does that make sense? So what that means is that, and I'm going to go back to where we were, hexokinase and a myosin head group both have an ATP binding domain. Are they going to do different things with that ATP? Potentially. But think about it as multiple proteins can carry out the same function, or part of multiple proteins can carry out the same function. And that would be known as a domain. Now, a structural domain is a little bit more, it's a little bit trickier. Structural domains are going to be, again, parts of a protein. What they are going to do is they are going to contribute to the overall structure of a protein and the stability of a protein. And a structural domain could be something like, well, there's a beta strand followed by an alpha helix followed by a beta strand or a beta sheet, beta alpha beta. You might have something like that as a structural domain within a protein. Now, that is a pretty common thing for proteins. To speak more generally, we can look at this example right here. GAPDH. This is a protein that's involved in glycolysis. This is glyceraldehyde 3-phosphate dehydrogenase, and it has two major domains. One domain is for binding a molecule known as nicotinamide adenine dinucleotide, and the other domain is for carrying out a catalytic reaction. So it's almost like if my two hands did different things. Well, this one picks up a baseball. Actually, yeah, my left hand is where my baseball mitt goes. That hand is a catalytic domain that catches. The other hand is, well, that's where I position my finger so that I can throw a curveball or splitter or anything like that. This is carrying out a different function than this right here. This one is just catching. This one is manipulating and throwing. Different catalytic domains, or sorry, different domains. They do different things. So different parts of a protein do different things. In addition to that, some of those domains are found in lots of other proteins. So this NAD binding domain, that's going to be found in lots of different proteins. And the sequence of those domains are going to be very, very similar to one another. Okay, now we've got a couple more slides for this and a couple of different ways in which proteins can be processed. Protein folding is important. A protein getting to its native state is very important. Sometimes proteins do not do that all by themselves. Instead, they might need some help. And that help might come in the form of what's known as a chaperonin or a chaperone protein. Or if you've ever heard of HSP 70, just as an example, or HSP followed by a number. Could you add a number to the chat if you've ever heard of HSP something? And a letter if no, you haven't. So HSP stands for heat shock protein. And essentially what these proteins are is basically they're proteins that are activated upon heat. And this is like relative heat. So I go out where it's 110 degrees outside. So heat shock proteins get activated. And then what they're going to do is they're going to help some proteins fold into their proper shape. So that's their function. Not all proteins are, or not all chaperones are heat shock proteins, but instead there are other complexes. For instance, here, GRO-EL or the GRO-EL-GRO-ES complex. Now, this is a protein complex that basically helps fold proteins. So what that means is that I'm just going to have a protein that I'm going to call A, and it needs to go from A to lowercase a. Okay, so unfolded, folded. Now, along the way, the mRNA gets to the ribosome, and at the ribosome we synthesize our protein, we translate our protein, and that protein goes from A, its unfolded state, and reaches A star. That A star needs to get to lowercase a. This is kind of a partially folded form of the protein. Okay, so our protein kind of gets hung up there. It's partially folded, it's not completely folded. And this is like without any sort of intervention. There are chaperonins or chaperone proteins that take partially folded proteins and produce the fully folded protein. So it's almost like they complete the folding of a protein. So chaperones are proteins that do that. They're more or less support proteins. They help with the processing of a protein. There are other proteins known as proteasomes. Proteasomes, has anyone ever heard of a proteasome? I think that might be a little bit more common, but what a proteasome is, and in particular, there's one known as the 26S proteasome. What this protein does is it's basically a kind of protein garbage disposal in some respects. What it's going to do is proteins, just like all substances, kind of have a natural life cycle. A protein will be tagged and sent to the 26S proteasome or a proteasome in order to be degraded. So it's almost like, hey, this protein's been produced, but we don't need it anymore. So we're going to tag it and then we're going to destroy it. Now, this next slide, I don't want you to get overwhelmed by this one because this is just the method or the mechanism by which a protein gets tagged and then destroyed. This is known as ubiquitination. Ubiquitin, if you've ever heard, or if you've ever heard the word ubiquitous, that means it's everywhere. Well, ubiquitin is a protein. It's a very small protein. And it gets added to proteins that need to be degraded, need to be destroyed. So when a protein or a substrate down here gets a bunch of ubiquitin subunits added to it, that's a signal that, hey, we're going to destroy this protein. So ubiquitination is the process by which you tag a protein for destruction. Chaperones help fold proteins. Proteasomes help destroy proteins. Okay, then, oh, does anyone have any questions at this point? I'm going to take a sip of my drink.
Quick question.
Mhm.
Just to make sure that I'm understanding it correctly, ubiquitination is the process of like IDing the protein that needs to be destroyed, but um, proteasomes is the actual process of doing the destroying. That's the difference.
Yes. So when a protein has been when several ubiquitin tags have been added to a protein, that protein is then ready to go to the proteasome to be chopped up.
So would it not be like, so does ubiquitin have to happen, the addition of that have to happen before the proteasomes act?
That's a great question. So the proteasome, that's a great question because proteins go through a couple of different life cycles, or there are different life cycles for different proteins. Some proteins will go to the lysosome, the membrane-bound organelle found within a cell, and that's how they are going to get degraded. And it has to do with the primary sequence of a protein, whether or not it's going to be ubiquitinated and destroyed or lysosomally destroyed. So the proteasome is kind of just sitting there. It's in the cell, it's ready to go. And just as the lysosome is sitting there ready to go, ubiquitination, well, it's dependent on a couple of different enzymes for adding that ubiquitin to a target protein. And they are certainly going to be those proteins involved in ubiquitination, they're going to be upregulated under some circumstances, whereas they might be downregulated or not particularly active in other circumstances. So if there's like a pathogen nearby, or if the cell senses like, hey, there's a pathogen nearby, that might up activate ubiquitination.
Okay, that makes sense. Thank you. I just was curious on like, they're just very similar. So I just wanted to understand like what they do when it happens. So thank you so much for answering that. I appreciate it.
Absolutely. Absolutely. Great questions. Um, so this is something that if you take metabolism, you're going to get more into that. You're going to get more into like, well, what's the process? how do we select a protein for lysosomal degradation, and how do we select a protein for ubiquitin-based or proteasomal degradation? And it has to do in part with the sequence of their amino acids. So those will be triggers for lysosomal degradation, but not for ubiquitination. Okay. If you ever see the IDPs, if you ever see those letters, that's in reference to an intrinsically disordered protein. So not all proteins fold into stable structures. Intrinsically disordered proteins, or IDPs, have regions favoring disorder. So think about random coils that, from one snapshot of the protein, which, sorry to kind of talk over myself, but whenever these proteins reach this native state, there's still fluidity, there's still movement of that protein. I always kind of think about movement of my lungs as being kind of the motion that a protein that undergoes. Like there's constant movement. It has a same general structure, but there's always a little bit of motion. Now, intrinsically disordered proteins, I always kind of think about them as, well, you've got this region that is a bunch of or that always leads to a random coil. And some of the traits of those regions is that they lack hydrophobic residues, which, take a moment and think about that. If you've got a bunch of hydrophobic residues, well, they might hate water, but they love each other. So they'll cluster together, and that's going to stabilize that. If you lack hydrophobic residues, then that means that you've got a bunch of hydrophilic residues, like your ionic or your ionic residues, or your polar positive, polar negative, polar uncharged. Those are going to love interacting with water, and they're going to love interacting with this water molecule, then that molecule. It's like taking my kids to Target. Oh, I want to go to the grocery area. No, I want to go to the snack area. I want to go to the toy area. These are rich in polar amino acids and proline. Now, proline is a non-polar residue, but what do we remember about proline? It's a problem because the proline R group looks like this. Here's my alpha carbon. I've got C, 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 boom. That ring structure is super rigid, super unforgiving. So that's going to kind of dictate. Oh, you thought you had some organized structure, like a secondary structure? Boom, proline says not so fast. IDPs may favor adaptation to binding another protein. IDPs may favor being modified. They may be more involved in signaling and regulation, and non-IDPs are more involved in catalysis and transport. So this item down here, signaling, might mean that they're a little bit more likely to be associated with a membrane so that they can communicate. Hey, this is what's going on outside of the cell. I'm going to transmit this signal from outside to in, and here's what we're going to begin in order to respond. Okay, now, good. Last, quaternary structure. Quaternary structure, multiple polypeptides are involved. Monomers only have a tertiary structure. Dimers and beyond have a quaternary structure. And whenever I say only have a tertiary structure, what I mean by that is the highest level of organization or structure that they may have. So lots of these proteins have terminology that you're probably familiar with: dimer, trimer, tetramer, pentamer. And if you're not familiar with that, you can kind of deduce that. Octamer, nonomer, and I could go on, but I'm going to stop there because nonomer is the highest that I can remember in sequence. Otherwise, I have to jump all the way up to like dodecamer. Um, the hydrophobic effect is a very big component. These hydrophobic residues love clustering together, aiding water. On the right-hand side, we have a tetramer. That tetramer specifically is hemoglobin. That tetramer, or that hemoglobin tetramer, is a perfect kind of transition to talk about, well, oxygen binding proteins. Now, one of the things that I want you to recognize about this particular tetramer is lots of those groups look very similar to one another. Lots of those different proteins, those different polypeptides, the purple, green, blue, and kind of olive colored proteins, You might have to rotate one on an axis or flip it, mirror it, or something like that. But these proteins are intended to look similar to one another because they are similar to one another. This is a good example of basically a homotetramer. Now, hemoglobin does have a structure that's a little bit more complicated than that. And in order to address that, we're going to go ahead and jump into our discussion of, excuse me, hemoglobin um, and protein function. So, what we've talked about is primary, secondary, tertiary, and quaternary structures, the organization of them, the implications of adding a particular denaturant. We can denature and renature a protein. Christian Anfinsen was instrumental in working on that. Chow and Fastman were instrumental in telling us that, hey, did you know that the primary structure holds everything that you need to get to the tertiary structure? There are, however, some instances of proteins that need a little bit of a push to get to their final folded or native state. And in order to do that, they're going to use something like a chaperone or a heat shock protein to facilitate that. Now, what we haven't really gotten into is what are any of these proteins doing? The GROEL, GROES complex, yeah, they help refold a protein. We're kind of taking for granted that that's a, a tetrameric complex that assembled. We're taking that a little bit for granted but now what we're going to get into is, well, what do these different proteins do? So we'll go into this a little bit. We're not gonna try to get through all 43 slides right now. This slide deck is on the Tuesday portion. It's underneath lecture recording on Tuesday the 3rd, protein function, hemoglobin, and myoglobin. There's also the PDF for that, which reminds me, I'm looking at the calendar on one of my screens that your problem set is due tonight by 11:59 p.m. And then we've got our second quiz on Thursday. Okay. And just as a little bit of a preview, the quiz on Thursday is going to cover yesterday's stuff and hemoglobin and myoglobin. Or sorry, it's going to finish with hemoglobin and myoglobin. Let me rephrase it for a third time. Finish with the end of hemoglobin and myoglobin. So if you're wanting to prepare for that quiz, download this slide deck, look through that, but we'll also cover it tomorrow. Will the exam have similar style questions to the quizzes? That's the plan. I will, however, tell you that I'm planning to have a couple of more images. For instance, I'll show you a picture of a peptide and maybe circle something and be like, what is this amino acid? Or what is the single letter code for this amino acid? But overall, yes, there will be a similar style. And that's just because the writing style that I started with in ninth grade is kind of what I've kept to now. Okay. So whenever we talk about protein function, there's a couple of different model proteins that people like to use. Sometimes someone will talk about protein function and they'll use immunoglobulins. So proteins that function by helping your immune system. A classical model, however, is to look at oxygen binding proteins. Now, with that in mind, our oxygen binding proteins, hemoglobin and myoglobin, are going to follow this, and they're a great model for this reversible binding of, well, a protein that binds onto oxygen. There are some important things to know about this. First and foremost, this is reversible. Hemoglobin can bind oxygen, and lo and behold, it can release oxygen. Myoglobin can bind oxygen, it can release oxygen. I say this in contrast to, our myosin head groups. Myosin head groups bind to actin, and they release it. But that myosin head group also catalyzed a reaction. And so that's why we don't really use that as a model for protein ligand binding. In addition to that, it's a little bit easier and more concrete to look at protein ligand interactions. Now, the terminology that we use to reference kind of binding partners is you've got a protein and you've got a ligand. If you're at all familiar with enzymes, that terminology is a little bit different. Rather than saying ligand, we say substrate. But when we're talking about something that is reversible and importantly, non-catalytic, we use the term ligand. Some people will say that as ligand, and they are wrong. Not really. I don't know. There's lots of people who have a really strong opinion about that. I don't. Um, so this is a reversible process. Protein ligand. Um, Ligands can be a variety of different molecules. We're starting simple, and O2 molecules are ligand, but they can be small organic molecules. They can be inorganic compounds. They can even be proteins themselves. And whenever I talk about a protein as a ligand, I think it's reasonable to presume that that protein would be something small, like 75 amino acids. Ubiquitin, for instance, is about 72 amino acids, I think. So it's a fairly small protein, but it's still a protein. Now, ligand binding is, most importantly, non-catalytic. It can be regulatory, and then this thing right here often causes a protein's conformational change. That term conformation. I want you to think about a conformational change as a shape change. So in one shape, it binds and then the protein adopts a slightly different shape. And that's always kind of tough to wrap your mind around because at this point in time, when we talk about shapes, it's not like, oh yeah, hemoglobin takes on the circle shape or it takes on a square shape. Instead, it's like there's an alpha helix and it kind of looks like a partially deflated football. Yes, that is the shape that we're referencing. So it is a little bit of a, I gotta wrap my mind around that. And maybe the term shape is a little bit of a stretch, so that's why people use conformation. But ligand binding usually coincides with or induces or produces a conformational change. Now, this is all about oxygen transport. In the lungs, we have a very high concentration of oxygen. In your muscles and in your tissues, you don't have a very large concentration of oxygen. But thankfully, we have a protein known as hemoglobin that takes our oxygen from our lungs to the tissues. So O2 binds and is transported to the tissues. Now, this takes place through the arteries. Okay, so oxyhemoglobin is the form that hemoglobin is going to take whenever it's fully oxygenated. Now, if you look closely at this model right here, what I want you to see is it kind of looks like a Trivial Pursuit piece, but rather than having like 8 pieces, it's instead got 4. That is meant to represent the 4 different subunits of hemoglobin. So hemoglobin is a tetramer. And that is the quaternary structure of a protein, of this protein. Each one of those can bind an iron ion. And it's Fe2 plus. That iron ion will help hemoglobin bind oxygen. That oxygen is going to be transported through the bloodstream and released in the tissues. And whenever that's released in the tissues, that O2 is going to go to myoglobin, or it's going to go to, what is that thing right there? I'll give you a second to type it in, or if you're on screen, you can do it. Absolutely, it's our mitochondria. That is the powerhouse of the cell because, well, O2 is going to contribute to aerobic metabolism. We can use oxygen to break things down, to get energy from them. So we want oxygen to be in our tissues. We want oxygen to be deposited to the mitochondria. We want a protein, myoglobin, to kind of act as a storage molecule within the cells or within the tissues so that, hey, it can hold on to a little bit of oxygen. So when it's needed, hey, it can release some oxygen. Um, but we don't want to overwhelm the cell with oxygen. So myoglobin holds on to oxygen, which oxygen is relatively uh, insoluble in a solution, or it's not super soluble in a solution. So myoglobin holds on to oxygen until we need it. And then that oxygen can end up in the mitochondria where we can help out with metabolism. One of the waste products of that, CO2. That CO2 we need to get from the tissue to through our bloodstream to our lungs so that we can exhale. Okay. Now, myoglobin and hemoglobin. These are proteins that carry out different activities, but they have similar functions. Myoglobin functions as an oxygen storage and transport protein in the muscles. Hemoglobin does the same thing, but in blood vessels. Myoglobin and hemoglobin are built on a common structural motif. Think about this as a structural domain. This is known as a globin fold. Okay, now with that in mind, you might think, well, what am I looking at here? So think about the globin fold or the structural motif as the organization of this protein. Now, this is myoglobin. The shorthand that we use for that is MB. It is a monomer. Over here, we've got HB, or hemoglobin. It is a tetramer. Now, if you look at, let's just pick out this one right here, the beta 2 subunit. We've got our N terminus, and we've got our C terminus of that individual polypeptide. We've got all these letters, A, B, C, D, E, F, and so on. What are those? Well, this protein is predominantly alpha helices. Or it's a bunch of alpha helices. It folds into a tertiary structure with a bunch of these alpha helices. So the beta 2 subunit is all of these alpha helices folded to give us that shape. Then all of these proteins fold in the exact same way, effectively, to give us this overall shape. Now, one thing that I want you to remember is that if you were to take this myoglobin subunit, and let's say that we rotated it and we tried to overlay that subunit on this right here, is there going to be some similarity? Yes. But one thing that I do not want you to make the mistake on is to think that 4 myoglobin subunits make hemoglobin. That is not the case. Myoglobin is its own protein. Myoglobin, as its own protein, exists as a monomer. Hemoglobin is 4 different proteins. Those 4 different proteins are very, very similar, and they cluster together to give us a tetramer. Now, at the center of all of these proteins is this right here. That is a prosthetic group. Here's one, here's one. Let me erase some of my other markings. There, and there. We've got 4 prosthetic groups. Those 4 different prosthetic groups are all heme, and what they do is coordinate an iron ion in the center of them. Now, whenever I think about that term prosthetic group, well, I think about, a prosthesis. A prosthetic group, a molecular prosthesis, basically, that enables a protein to do something that the amino acids alone cannot do well. So, heme, this molecule, helps this protein, myoglobin or the hemoglobin subunits, they help them do something, and that something is to bind O2. Without heme, myoglobin and hemoglobin, they're not going to bind O2 very well. Can they do it? Maybe, but it's not going to be a very strong connection. Whereas with heme and the iron ion in the center of it, they're very effective at binding O2. So if we look at the next slide, and this next slide is where I'm going to stop it for the day because I feel pretty good about that. We've got myoglobin. This is a different structure, a different depiction of it. We've again got the N termini and then go all the way to the C termini here. If you look closely, it's kind of hard to see, but that's 153. Myoglobin is approximately, or most myoglobin sequences are about 153 amino acids. They might vary ever so slightly, but not radically. We've got the heme prosthetic group right there. At the center is our iron ion. That iron ion is Fe2+. Now, can iron take on different oxidation states? Yep, it absolutely can. Fe2+ can bind O2 very well. Fe3+, not so much. So there's a preference for that Fe2+. This is myoglobin, which is approximately 90% alpha helix. All the hemoglobin subunits are also approximately 90% alpha helices. They will bind O2, which is projected right here, and amino acids in this part of the protein and in this part of the protein will help out with the coordination of that O2. Okay, so this is where I'm going to stop for today. Myoglobin, we're going to get into heme tomorrow. We're going to get into the rest of this protein complex and these proteins and how they function, how they bind O2, how they release O2, and everything like that. We've covered a lot of material, but keep up with your learning objectives. Or keep up with looking at those learning objectives and trying to say like, okay, do I have an answer for this? Can I formulate a question on this? And I think that would be a really helpful and productive thing for everyone. But also, I don't have office hours tomorrow, but I'll have them again on Thursday. Same thing. If you want to meet tomorrow at some point, just send me an email. I can accommodate that. I'm going to stop my recording now. If anyone has any questions, they're more than welcome to hang around and ask them. Other than that, though, I hope you guys have a great afternoon. And thank you very much for coming to class. So I will